بسم الله الرحمن الرحيم ربنا آتنا في الدنيا حسنة في الآخرة حسنة وقنا عذاب النار. So tonight I'll be speaking about neuroscience, vision and mapping. So simply speaking, we'll be discussing the concepts of receptive fields and projective fields. But first I will begin with mentioning that I came across this particular paper, Why are what and where processed by separate cortical visual systems, computational investigation. So I looked at this particular experiment here, as you can see, and trying to unearth and understand the model used here, which we will go into in a moment, and also understand how various fields, in particular receptive and projective fields, are being investigated to understand the concept of space, object recognition, mapping and navigation. So we'll begin with this particular textbook, which I really like, by Samir Zeki, Turkish neuroscientist. And in this particular text he speaks of vision from the retina to the neurons to the systems to the networks, essentially covering everything. But we'll go to chapter 9, which is of relevance today. And here I am going to speak about receptive fields, so we can understand this in much more detail here. Right, so
is it a problem, goddamn what?
hello? Can everyone hear me? Can everyone hear me? Can everyone hear me? Right. Okay. I'm just trying to adjust the audio. Just give me a second here. Hello? I'm just trying to adjust the audio. Just give me a second here. Right, so everything is going good. So I can tell. Right, so first and foremost, we have to understand that the vision system in the brain lies in various places. Today we're going to focus on V1, typically speaking, it's around this area, which is the posterior region of the cortex here. Okay, and going back to the paper I spoke of, this particular experiment was aiming to look at how mappings are divided into simpler mappings when a stimulus is presented to the eye, in this case directly at the retina. And the examples given here were various shapes. Now I must also mention that this is all theoretical. There can be errors, of course, and much of this is typically agreed upon in terms of the nature of how these experiments are conducted, nature being understanding the root representations of the brain when it comes to images, shape, form, shading, so on and so forth. I actually came across this particular experiment when reading a book by Coslin called Image and Brain, and I find his work quite fascinating on images and visual networks. So, the biggest issue I have is trying to understand receptive fields and projective fields. So that's what we're going to concentrate on. Okay. And Samir Zeki speaks of this as well. So just again to reiterate, keep the V1 area in mind moving forward. So here's a GBT that I created a few years ago called Brain Explorer. You can obviously download it and use it. A receptive field of a neuron is the region of the visual field or retina in which a stimulus must fall to change the neuron's firing rate. So a neuron firing rate is where a neuron changes its behavior as per the stimuli presented. This could be in the context of visual neuroscience, saccade, could be rapid visions, it could be images or videos, it could be physical objects in the real world. In any case, various firings are presented, and these firing recordings are similar to how the neurons in the motor cortex behave. And there was an interesting experiment done with monkeys moving their arms in order to understand which angles fire neurons the most. I'll very quickly show you this particular experiment. This is a fantastic resource online by McGovern Medical School from Texas, and to give you an idea of a neuronal firing, here we go. So if I was to move this hand, for example, 135 degrees, you can see the neurons firing quite high. That blue area, just to double check if it's coming up on the screen here. Right, it's not coming up on the YouTube streams, just bear with me here. Let me just change this. Okay, so should work now. So what I did was essentially press 135 degrees here to demonstrate neuronal firing. So I'll show you one more time. This is an experiment done in the 80s by Georgopoulos, looking at monkeys and how neurons are fired. I'm just giving this example, which is loosely related to visual neuroscience, because the motor cortex, i.e., movement of your limbs, is related to the vision system, of course. So again, 135 degrees, you can see a high level of firing of neurons right over there. However, if I move this to the 45-degree mark, you'll see less difference. Now, why such a big discrimination between the two? That's a completely separate topic. This is a different area of motor cortex, but this is just to demonstrate how neurons are fired. Again, show you one more time. 225 degrees, you can see some response there, and if I go to 315 degrees, you can see no response almost. So it's a fascinating area that baffles many neuroscientists, and okay, right, so this is done. Okay, so yeah, with observing all of this, we can see the nature of neuronal firing. So going back to this particular paper, the idea here is that essentially the weights are adjusted on the connections that come from this input layer, which is seemingly the retina, going into a hidden layer. Okay, that particular hidden layer then leads to the output layer, which then confirms which neuron is processing which side of the stimulus presented. Right, so by analyzing the patterns of weights on connections to and from the hidden unit here, it could be ascertained how mappings are performed. Now, by looking at which connections from the input layer contribute heavily to the activation of a given hidden unit, we could determine which aspects of the input were particularly important for that unit. This is called analysis of receptive field. Now, this is what a receptive field is. Looking at the analysis of the shapes presented here. Okay, so to give an analogy, this is the relationship between a neuron, for example, this image presented to one of these neurons, whichever it might be, okay, and the area of space in which the stimulus can drive it. Now, the receptive field of a hidden unit consists of those input units that have strong weights on the connections to the hidden unit. Hence, what that means is essentially that influence is strong when stimulated by an input. So, when an input is stimulated, it seems so, quote unquote, naturally, that these would also be stimulated, right? So this is again looking at how vision is recorded in the eye. Okay, so now we're gonna move on from here by examining which connections from a hidden unit contribute heavily to the activation state of output units, and one could determine which combinations of input properties as registered by the hidden unit. So now it's not just one single aspect of this whole thing. Now it's a case of looking at obviously how various inputs, 1, 2, 3, 4, 5, however many, have an influence here, right? Where so this idea of combinations of input properties is projective fields. So you have the receptive fields, okay, looking at those inputs that are important, and then the combination of said important inputs supposedly for specific responses. This is called analysis of projective fields. Now, the projective field of a hidden unit consists of those output units that have a strong weight on their connections from that hidden unit. So this is what I would describe it as. Again, this is just what I think would be this case, right? So we can say in this case, for instance, so 1 second, what's going on here? Okay, so this is what I'm trying to say here. So if I just grab this, actually grab this and scribble. So for example, hidden unit, as we can see here, H, this is wrong. I need a free flowing thing here. Okay, so what I'm saying is this essentially HU equals, right, hidden unit possibly equal to the weight, which is W, okay, the weight and that weight which we are speaking of here is determined by a factor of unknown, and here n has to be more than 1, I would say. Okay, this is really annoying. N is 1. So this is what we're looking at here essentially. So yeah, it's quite fascinating to understand this. Now this is really advanced, right? And this is something that requires a lot of heavy thought, so to speak. So let's just read the introduction just so it summarizes what exactly they're looking at here. So I get rid of this thing on the left here. Here we go. Okay, so abstract. In the primate visual system, the identification of objects and the processing of spatial information are accomplished by different cortical pathways. Spatial information here is key, okay? Because the information of things around us in a confined space, i.e., our environment, is processed according to these authors by different cortical pathways. Now, the computational properties of this two systems design were explored by constructing simplifying connectionist models. Now, connectionists believe that a unit corresponds to an area on the neuron, which therefore leads to an output which can contribute to a system/network, rather than taking the whole neuron or the entire area as a whole, the specific regions or areas, right? So the models were designed to simultaneously classify and locate shapes that could appear in multiple positions in the matrix, and the ease of forming representations of the two kinds of information were measured. Some networks were designed so that all hidden nodes projected to all output nodes, where others and the hidden nodes split into two groups, with some projecting to the output node that registered shape identity and the remainder projecting to the output nodes that registered location. The simulations revealed that splitting processing into separate streams for identifying and locating a shape led to better performance only under some circumstances. Provided that enough computational resources were available in both streams, split networks were able to develop more efficient internal representations as revealed by detailed analysis of the patterns of weights between connections. So what this is saying is that networks, some of them were designed so that all hidden nodes essentially landed to all input nodes, where hidden nodes split into two groups, seemingly. Going back to this image here, seem to split into two nodes, right? And this allows us to at least understand from this level here or this level here, where are these units connecting with each other to give the output that we're looking at. And the hidden layer is the biggest mystery here, not because it's hidden layer necessarily by name, but the nature of how it's behaving when it comes to processing the images. So that's what my understanding is so far. So with some projecting to the output nodes that registered shape identity and the remainder projecting to the output nodes that registered location. So the shape was identified and the location. Now these are very, very, very important aspects, especially when it comes to navigation and vision. Identifying shapes and identifying the location of the objects and the shapes you look at allows us to navigate and move in this world. Okay, and an interesting paper that we'll discuss on the next stream is this one here, which is it's an article actually. It's here somewhere. Here we go. So this article is quite fascinating because, as you can see here from this graphic, quickly download and show it to y'all. So this is what I'm working towards, inshallah, moving forward. So this allows us to give more context to why these imaging systems are necessary to understand. So, very briefly speaking here, the hippocampus, as you can see here highlighted in the orange portion, and the entorhinal cortex, which is a small bit here labeled D. This hippocampus is labeled A here. This allows, in this case, a rat to navigate, right? And this in and of itself is not enough, because the rat must also identify shapes and identify distance and direction as shown here, right? So this is something that was discovered about 20 or so years ago by the Scandinavian researchers Moser, known as grid cells, and they look at location and direction. So it's a natural GPS system we human beings have, which is quite more remarkable, right? So this article we shall be looking at, inshallah, on the next stream. But for now, I just want to lay the foundations here with this paper I came across, and of course allowing you to go back, and really ponder and dig deep as to how Allah has created such a complicated system. That one perspective I'm giving you is a connectionist perspective, a computational neuroscience research paper here, right? Which I'm still learning, mind you, but the theory side of things is something that I find fascinating and a strength of mine in. Anyway, shape, as you can see here, and location. So these are various representations that may match the 5x5 grid here. So 5 y-axis, 5 x-axis, okay, and each shape represents a location, and each location is pointing towards a shape. So they're in tandem, I would say here. Okay. So the simulations reveal that splitting processing into separate streams for identifying and locating a shape. So now you have two separate streams. One is locating, one's identifying the shape, led to better performance only under some circumstances, provided enough computational resources were available in both streams. So now, if there were enough resources, whatever that might mean, split networks were able to develop more efficient internal representations, as revealed by detailed analysis of the patterns of weights between connections. Now, before we go into shape, form, shade, and location, let's understand the fields upon which these representations are seemingly falling on. So let's read Zeki's text here. This is the first time I'm actually also reading this particular chapter on the visual system. I was reading Zeki's work when looking at fixation and attention, which is another really interesting area. But anyway, hierarchies in the visual system. The notion of hierarchies in the visual system was to have a powerful negative influence in questioning the doctrine of functional specialization in visual cortex, and hence of separate pathways and areas for color vision. This is surprising because nowhere is the doctrine of hierarchies adequately formulated in physiological forms. It nevertheless permeated thinking and created an attitude of mind that was prevalent among many physiologists, at least for a time. Its influence has been due not so much to dismissing the evidence in favor of functional specialization, similar term used in computational neuroscience when explaining systems and networks, but rather to ignoring it and supposing thereby that the hierarchical strategy is the sole strategy that can account for the operations of the visual system, but an exclusively hierarchical strategy which implies the visual areas are connected strictly serially with each other, is incompatible with the observation of specific visual defects which affect selectively certain attributes of vision. Example given: color vision or depth perception. Now, the idea of color perception, may I add here, and depth perception are two separate concepts completely. Okay, depth perception is the idea of how far or close an object is, for example, and deception, to use a term here, is usually the case of depth perception being miscalculated, for example, in the experiments of visual cliff. Okay, and color vision, obviously, you have individuals who have issues with not being able to see green, for instance, or their grayscale comparison fails them. So, since a lesion in any of these visual areas should compromise all the attributes of vision more or less equally, because all the attributes of vision would be expected to be represented in each visual area. This consideration did not present a problem. Such observations had been successfully dismissed and did not therefore pose a serious obstacle, assuming anyone to have been aware of them in the 50s. Again, in the 50s, you had Hubel and Wiesel, who did work with cats, in fact, and their visual cortex. And I think it was quoted on this paper somewhere. Let me do a quick search here. No, okay, no problem. So, moreover, there were excellent reasons for supposing that the hierarchical strategy may be the sole one that the brain uses to analyze the visual environment. So, the origin of the hierarchical concept. As with so much else that was to mold the thinking of neurologists, it is difficult to trace the origin of the concept of hierarchies and visions, but central to it is the usually unspoken assumption that objects in the visual world are conveniently labeled, and that all that the visual system has to do is to analyze that label. The analysis can be undertaken hierarchically, with each level of the visual pathways undertaking an analysis at a certain level of complexity, and leaving it to subsequent stages to continue the analysis at a more complex level, or at least to analyze more of the label. What does that mean? So, assumption, unspoken assumption, this is a key term, and I will highlight that here, that objects in the visual world are conveniently labeled, and that all that the visual system has to do is analyze that label. Is that true? I would say there is an a priori assumption that most of what we tend to know and have learnt as we have matured and grown up is that things are labeled. For example, how do you know a horse is a horse? Okay, how do you know a table is a table? So these are aspects that really confuse neuroscientists and, of course, philosophers alike. So this idea that things have an unspoken label is not agreed upon, and this is why science fails on so many levels when it comes to metaphysics and providing clear definitions of how we are to function in this world. Okay, the analysis can be undertaken hierarchically, with each level of the visual pathways undertaken an analysis at a certain level of complexity, and leaving it to subsequent stages to continue the analysis at a more complex level, or at least analyze more of the label. Simply speaking, the analysis, whatever the analysis is, it could be computational, it could be neurobiological, it can be studies around lesions that neurologists work on and neurosurgeons obviously operate upon. Whatever the analysis may be, Each level of said visual pathway has a certain level of complexity, obviously, I think it's quite obvious, and leaving it to subsequent stages, whatever in the future, in other words, that the analysis will be either more complex at that level or more complex at different levels, or at least that label is analyzed at a particular level. So, computational neuroscience really is trying to look at how these aspects of the world, objects, the dunya, is being analyzed by our mind. And it's very difficult to have a universal agreement. This is why the Quran speaks about going out and looking around and analyzing the world and the objects around us, right? I started this stream with this paper and I'll go back very briefly. This experiment here, which is trying to explain how this image and this T like object and its location is being interpreted by our retina, which goes through 3 stages of analysis, supposedly, and how each neuron is going through, each neuron visualizes and therefore processes a location and also identification of shape. So, a developmental psychologist, for example, they would say, well, a child learns this because it has innate information. An evolutionary psychologist or evolutionary biologist would say, well, we developed this because of our ancestors and so on and so forth. But in all 3 situations, there is no universal agreement as to how we are able to recognize and identify. So this is one of many complex quote unquote systems that Zeki speaks of here. Okay. Right, so moving forward, no one should underrate the importance of unspoken assumptions. They are often the most powerful because they are part of the scientific culture and therefore remain unquestioned, though they get perpetuated. So what that means is various rules and laws are unspoken. For example, the idea of a solid being a solid, a liquid being a liquid, a gas being a gas. No one really sits and debates solid, what's S, what's O, what's L, what's I, what's D. It's just something that exists. It's part of the lexicon. Moreover, with an attribute such as color, there was at least a plausible reason for supposing that there is a convenient label rooted in the belief that color of an object is determined by the excess of any given wavelength reflected from the object, that a green object looks green because it reflects more green light. So this is interesting because he uses the term belief, not rule, not law, not even theory, belief. So what distinguishes belief from evidence, for example? Elliot Sober speaks about this, and I'll be very brief here. Belief is the idea that something that has happened so many times over and over again that it's now become belief. Similar to a fact, I would say in this case. Some philosophers, I think it's Searle, Searle, would disagree. People who are very reductionist would say that, well, fact is not the same as belief. A fact can change under other circumstances and not be a fact anymore, but a belief remains true for that individual or group of people. In any case, the idea that an object seems green because of the green light that is reflected is something that is still believed today. When we look at the sky, the idea is that the sky is blue because the blue light that comes out from the white light spectrum is being reflected the most on planet Earth. Anyway, and to repeat, it is useless to plead that there were some who had seen the color is a vastly more complex problem than such a naive view would suggest. Those who had seen the mysteries of color vision had been remarkably inarticulate in communicating their enlightenment to others, even in the scientific community. Right, so this is again challenging views of beliefs within the scientific community. Now, this is not of interest exactly. I just gave you the idea of complexity and, of course, hierarchies and how they exist, right? And our main focus for tonight on this stream is the visual system, and more specifically the receptive field, which we spoke about earlier. Okay, so the physiological evidence for hierarchies. Yes, hierarchies do exist within our system. Okay, so using the cat and the monkey, Hubel and Weisel had been studying the physiological properties of cells in the primary visual cortex, which for simplicity we can refer to henceforth as V1. At the start of this stream, I said please keep the the region of V1 here, which plays an influential role in our visionary system. And I also mentioned earlier that Hubel and Weisel, you were studying cats, and for those interested, I'll very quickly pull up the reference here. I actually looked at this in Popper and Eccles' book. I have a physical copy, but the digital one should be here somewhere. Yep, here we go. Very quickly, I'll show that here. Should be here. Okay. Here we go. So this is what they were looking at in the cat's visual system. Okay, so when light enters into the eye and changing the orientation of the slit, there's a small gap between two stimuli, and how lightness and darkness and contrast has an effect, essentially, right? So this is what Hubel and Wiesel were looking at, essentially. Okay, so this is an experiment from the 60s. You can feel free to look at this. I highly recommend it. It's very interesting. So going back here, this can be done by recording the electrical activity of cells by means of electrodes inserted in their close neighborhood. This is done on cats and monkeys, and it's completely ethical and fine. Back in the day, they would not really take much care. Nowadays, they do. Again, defines ethics. Anyway, cells in the nervous system increase or decrease their ongoing electrical charges in response to appropriate stimulation. So this is at a very basic level how cells in the nervous system operate. These changes can be picked up by an electrode and fed into an oscilloscope and an audiometer. Oscilloscope is a device that measures wavelength of electrical activity. Audio monitor gives that, I think it's called silent noise, that picks up electrical activity. Now, visual stimuli are usually flashed on a screen facing the animal. Okay, and the part of the screen which when so stimulated yields a response from the cell is called receptive field. So when something's stimulated and a response comes from the cell, that's called a receptive field. So that field is being receptive. Here's an example. Okay, very simple basic example. RF is the receptive field. So imagine this is your eye, not the square, but from here to here, and the measurement reference here is 1 degree. 1 degree meaning the number 1, for example. Yeah, so here you have this patch is your receptive field. That's your fovea right here. Now with each orientation of light passing on the receptive field, the oscilloscope is recording different recordings. So this, for example, direction is more stimulated, and this one as well. These two are far less, right? In fact, there's a slight difference if you can spot it right here. This one is showing some drop in activity around the middle. This one isn't. So spotting these these differences is extremely important when looking at the visual system. So once the receptive field is plotted, one can study in great detail the kind of visual stimuli which, when flashed into the receptive field of the cell, give the best response. Hubel Weisel were not the only ones to undertake such studies. Others had been there before, but others had labored under the common but understandable misapprehension that the cells of the visual cortex would naturally respond to light. This is another thing. Not all parts of the brain that are responsible for vision pick up light. Some pick up data, you can say, through object, through shape, and some are responsible for vision when it comes to looking at nighttime. For example, the substantia nigra. Very quickly, I'll show you the substantia nigra and the, I think it's the superior colliculus night vision. Quickly show you here. Yeah, superior colliculus. By processing dark stimuli and using GABA, which is excitatory neurons, to regulate wakefulness in response to darkness. So superior colliculus is a very interesting looking part of the brain. It's just above the brainstem actually. So if I go here you go. This is the superior colliculus right here. You have the inferior, which is at the bottom, but what's interesting is the brainstem and the superior colliculi are not far from each other, because the medulla oblongata here is responsible for processing motion, and right above the superior colliculus is the basal ganglia, which we will speak on another stream. Basal ganglia is also responsible for decision making, and is also responsible for comprehension, so linguistics as well. And look how close the decision making and wakefulness is to the visual cortex right here. The cerebellum also plays a role, mind you, in decision making and motion, but for now this is just something I wanted to mention here. Quite fascinating. Right, so moving forward is the part here we go. I want to speak about this one as well. Concentration of light on a region. So quick to spot an important finding. Hubel and Weisel needed no such lessons. They were going to explain blah 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 blah. There we go. In Germany, Richard Jung had developed in the 50s one of the most advanced systems then available for recording the visual cortex, but because he used diffused illumination to activate his cells, he obtained little response from them. What does that mean, diffuse? So illumination that will happen and go away. I believe I could be wrong here. Is a lighting technique that uses a broad scattered light source to evenly illuminate a large area, providing a general overview of an object. Right, so it wasn't specific, it was scattered light all across one area. Right, okay, interesting. He obtained little response from them. Technologically, his approach was powerful. Conceptually, it was weak. Later in life, Jung was to blame others and his admin duties for not realizing that his stimuli were conceptually ineffective, advising young scientists that one should not leave the lab during a productive period of research. He also had this important advice to give that a field should first be explored by simple, qualitative pilot studies to find out the most rewarding approach before experiments are reduced to quantification with a single method. Yeah, I agree. You have to do a trial experiment, explore how the methods can be of use and if their utility is of any value way before you proceed with using them and having a quantifiable methodology and therefore having a conclusion which may not be what you expect, which is fine, but usually it would lead to failure. It seems so. Do I have a question here? Okay, so he recounts how Hartline had told him they had searched for the frog's off neurons by moving a stick through a diffuse light, and that when Jung proposed stimuli after chance. Running into a crack in the slide which drove a single cell in the cat's visual cortex. While without recourse to any complicated machinery, they exploited this finding by shining lights of different orientation. Here we go. On the screen facing the animal. At any rate, during the course of their studies, they discovered that all cells in V1 had a very small receptive fields, and that all cells outside the layer that receives the predominant input from the lateral genicular nucleus, which is LGN, which has various layers, were orientation selective. Quite fascinating. In fact, I first came across this in this book as well. No, I think I have a picture here. LGN. Oh yeah, here we go. So this is a picture of the LGN. So you have the right one and left one. So the left eye connects with the right connects with the left, and Zeki was speaking of layer 4. Should be layer 4, yeah. So layer 4 receives input from parvocellular, which is a pathway in the contralateral, which is the left side, and ipsilateral, which is the right side of each eye, right? So you can see again how from your eye down to the LGN, down to the recognition of objects, location and shape, they all have a complicated connection with each other. Anyway, such cells do not respond to diffuse light and respond only weakly to spots of light. To obtain an optimal response from them, the activating stimulus must be a line of the right orientation and must fall within the excitatory part of the cell's receptive field. That's key here, because if that's not happening, then your experiments are in vain. And what's the point? When you are shining light contralateral, which is the left side, or ipsilateral, which is the right side, and you're not targeting the receptive field, how are you going to determine if there's any lesions, for example? Or how are your experiments going to be of any benefit if you are unable to recognize the shape, the color, and location, and to a large degree the shades of the object? So this is very, very important. And this might seem arbitrary right now, and this might seem a very basic thing, that yes, of course, these fields have to be hit, but back then it was quite difficult to do so, right? And this is just giving you an idea, an insight into how neuroscience has progressed, because with each neuroscientific paper you will come across in textbook, it's important to understand, I want to say, the gradualism of a theory, but the gradual changes and how things have progressed from point A to point B, and from point B to point C, or point C to point J, and so on. The simplest of such cells were called, appropriately enough, simple cells. Different cells have different orientational preferences to give an optimal response. The appropriately oriented line had to extend the whole length of the cell's receptive field. Halving the length of the line falling within the cell's receptive field half the response. This is quite logical as well. Okay, very logical to think this as well. If a line is identified in a receptive field, and upon that line 25%, 30%, 100%, 200, well, not 200, sorry, 90% is being activated, then it's pretty simple and straightforward. Certain cells do not behave in that way. In fact, most cells, I would say, have their own properties and own working. So again, science does not explain everything in that case, right? Right. So, on the other hand, extending the appropriately oriented line beyond the boundaries of the receptive field did not alter the vigor of the response. Vigor of the response, strength. In addition, there were distinct subcomponents of these receptive fields. In particular, when a line of the appropriate orientation was flashed in a given region of the cell's receptive field, the cell gave an on response. When a line of the appropriate orientation was flashed in a given region of the cell's receptive field, the cell gave an on response. But flashing the same light in flanking regions not only inhibited the cell, there was a response when the stimulus was taken off, an off response, itself the direct consequence of preceding inhibition. It's quite fascinating, actually. epoch, making sure first that light was a necessary but not sufficient condition for activating all cells. This may seem obvious now, it was not so obvious before. Next, it showed, even if indirectly, that an image of the visual world is not impressed upon, the retina was as was commonly supposed. Those who commonly use this term were never explicit as to what it meant in physiological terms. Instead, such analysis as is carried out by the retina was known to be relatively simple, and it turned out that of the striated cortex was not that much more sophisticated either. Striated cortex is a thing near the temporal lobe, or just behind the temporal lobe, I think. Striated cortex, yeah. This V1, V2 area, that's correct, yeah. Here we go. This is what we're looking at, essentially. Occipital lobe. Okay, it's interesting. And instead of lastly to underline the important hierarchical principles for the properties of such cells could be accounted for, we will largely believe by supposing that each simple cell received convergent input from several lateral Augean cells, which would constitute the building blocks from the response of simple cells could be built up. Interesting. Okay. The most common feature of geniculate cells, like that of the retinal ganglion cells, is a center surround receptive field. Light falling in the center of the receptive field excites the cell, thus increasing its electrical discharge rate. Light falling in the surround inhibits the cell. The cell now increasing its discharge rate only when the light is flashed off. So these are the diagrams that will help us understand a bit more. So these are your neurons here. These are the receptive fields being shown on the eye. Okay, so on, off, and the orientation of the light. So a simple orientation selective cell behaves as if it receives input from several centers around antagonistic. What does that mean? Action cells of the lateral OGN-A, flashing a line with an orientation that stimulates more of the excitatory centers plus will stimulate the cell. So all these plus regions you can see, these are going to produce the neuronal firings that we were speaking about earlier in the stream. Okay, so yeah, so these circular regions you can see here, these are where the visual field is, okay? And it's quite fascinating understanding this. Okay, so fascinating stuff. So within this receptive field, light shines, you get stimulation. Sorry, electric discharge. Changing the orientation so that more of the inhibitory, which is a decrease, surrounds are stimulated will diminish the cell's response. An orthogonal, which means 90-degree orientation to the one shown in A. So our 90-degree change. So light, so this is a receptive field here. Light is going this way. So it's passing through all the positives. But if you turn in 90 degrees anti-clockwise, and then there's no response here. A will yield no response from the cell C, since it will invade both excitatory and inhibitory regions. So now it's both, which will therefore cancel out. However, B, it will diminish cell's response. So it will show a decrease. As you can see here, there's been high, and then it slowly goes low, and then it's gone away. Okay? That's how it works, essentially. Yeah, I think I came across this as well in Popper's book from the 70s. Just give me a second, I'll be back. Right, so we are back. So yeah, this is now the cells that exist within the retina. Give you an example of, for example, look, a receptive field is this middle bit, and the surroundings are this pink bit. So you can clearly see electrical discharge is way different in each case. So the cells of the inner layer of the retina, whose fibers project to the brain, of course, and the cells of the LGN do not respond to light as such. Rather, they respond to the difference of intensity of light between one part of the receptive fields to the other. So if you shine light on the entire field, there's no response. However, if light is shone on the particular receptive field, so rather they respond to a difference in the intensity of light between one, thus stimulating such cells with diffuse light A is ineffective in eliciting response. Flashing a light in the center of the receptive fields yields an on response, while in the surround a response is obtained only when the light is switched off. The cell is therefore said to have an antagonistic center surround or a concentric receptive field. So concentric meaning this, essentially. Okay, this is what we're looking at when understanding the visual system. Barlow and Kufla. I think I've heard of these researchers, if I'm not mistaken. Let me quickly check. Right. So yeah, going back here, we're going to look at. We shall call them Kufla-Barlow cells. So these are cells discovered by Barlow. Interestingly, he said that units are what corresponds to us thinking, not the brain as a whole, I believe. Interesting. The important point here is that their receptive structure is such that they could be said to constitute the very elements from which simple cells of V1 could be built up, thus reinforcing hierarchical doctrine. Interesting. So all these cells come together here to eventually concentrate in this area over here. It's quite fascinating. This particular area here. As mentioned, this area, the striate cortex, responsible provision right here. Right, so for those who have just joined, thank you very much. This is my GPT called Brain Explorer. You can download and play around with it regarding the neuroscientific knowledge you're looking to attain. So we looked at this projective fields. A projective field is less commonly discussed but conceptually simple. It is a set of downstream neurons that a neuron sends information to. If the receptive field tells you what the neuron sees, the projective field tells you whom the neuron talks to and influences. So this is where this part right, so this is done. Projective fields, let's look at projective fields. Okay, this will be an interesting idea. So it is downstream neurons. V1 neuron may project to V2 neurons tuned to similar orientation. Ganglion subprojected to LGN. This projective field defines LGN neurons activate. Computational meaning, pattern of output weight similar blah. Why both concepts matter in visual mapping. Receptive, determine which features. Projective fields determine how. Organized in one sentence. The pattern of so receptive field. Just to summarize, the pattern of inputs a neuron or unit responds to biologically a region of visual space, computationally pattern of weight by weights. Projective field, the pattern of downstream targets a neuron or unit influences biological. Okay, so now we're gonna go through projective field. I think receptive field has been covered nicely. Okay, so bookmark these as well. Bookmark this as well. Projective field. Okay. Projective field. Yes, this is organized here. I guess we can expand. Let me take a note, inshallah, on the organizational structure and nature of receptive fields. We'll do that next time, inshallah. No problem. For now, we'll look at projective fields. Okay. So I want to show, no problem. Right, okay, so in visual neuroscience, projective field is a set of neurons that are single neuron positive, while its receptive field is a set of neurons. Interesting. So Amarine cell. So wider projection. Okay, interesting. With extreme, we can break down the Amarine cells. Wow, subhanallah. Right, so projective field. Just trying to see what can I show that can best describe this. Okay, so here's one thing I found. I can read. Okay, so let's read this. Projective fields of cortical neurons. Okay. Actually, let's read this in the next stream. I think what I'll do is break down this myself and then discuss in more detail. Right, perfect. So, inshallah, hopefully, you have learned something of value. Inshallah, see you on the next stream. Assalamu alaikum wa rahmatullahi wa barakatuh.